And as mentioned, my name is Ali, and I'm responsible for the medical affairs function within Vigil. Today, I have the pleasure of really on behalf of many investigators and their teams presenting interim findings of the first natural history study in ALSP. And this data is current as of October, November of last year, but the study is ongoing. And really, before getting into the data, just overall, this is going to be just one slide on the disease to just warm you up. Background, I'm sure there are many people here who know a lot more about ALSP than I do. But nevertheless, just to have a standard setting for everyone, and then we'll get into sharing data specifically in biomarkers, MRI, clinical assessments, and a little bit of the early work that our translational medicine team is doing to correlate some of the biomarker findings with actual clinical outcomes. And again, before getting into anything, our sincere thanks to the participants, right? So it's a very selfless act to enroll in a natural history study in a disease such as this, both by the participants and their caregivers. But again, it really highlights the importance of, if we don't do this, I think the Orchard example was a great example, right? They did a study, and they referenced the natural history study to with their regulatory interactions. So a little bit of background about the disease: adult onset leukoencephalopathy with axonal spheroids and pigmented also referred to as CSF1R related disorder. Thankfully, it's rare. It's this autosomal dominant disorder caused by loss of function mutations in the colony stimulating factor 1 receptor gene. It accounts around 10% of known adult onset leukodystrophy. So it's maybe not as rare as we would like it to be. The group out of UCL led by David Lynch have it around 17 to 20% based on their experience. But if we go with what's been reported in literature, around 10%, it gives us around 10,000 patients potentially, right, in the United States. Thanks to the efforts of Dr. Bonkowski et al., as of last October, we have an ICD-10 code, very helpful and appreciated. With regards to symptoms, mainly patients present with either motor or cognitive symptoms, and a profound and progressive progression of these symptoms, where you will see most of the time incapacitation within 3 to 4 years, and unfortunately death shortly after. Now, since it's heterogeneous, cognitive and motor symptoms, many things go under that. As you could imagine, a lot of misdiagnosis or a lot of effort from our side and your colleagues in the field, as some of you are here, actually goes into educating. And where we see most of the misdiagnoses, both as we are out in the field and also where we're looking at retrospective analyses, these happen in FTD dementia clinics and also in MS clinics. So those are two areas where we're actually reaching out to increase awareness. Illuminate, as I mentioned, incredibly important study for the field. Really the main objective to characterize biomarkers, MRI, and clinical measures of disease progression. And it has a potential to serve as a synthetic control arm for interventional trials, and I emphasize trials. So we have a phase 2 study that's complete enrollment, so it's ongoing. So it's just one study, right? But really the hope is not just for us, for others to continue doing maybe open label, non-placebo controlled trial studies, and hopefully we'll be able to use this natural history data that's, again, will be gifted to us so selflessly by the individuals who are participating, enrolling up to 60 patients over 3 years. It's a global study, US, EU, and as of this month, also South America, specifically Brazil, is enrolling as well. And of course, the key assessments, we're looking at MRI every 6 months, CSF biomarkers baseline and every 12 months, and clinical assessments such as MoCA at baseline and also every 6 months as well. Eligibility criteria, we always mention this as we're talking to investigators. It's really the main reason we came up with this is based on actually the diagnostic criteria published by Kono et al., where they say if you have 3 or more symptoms, confirmed documentation of genetic mutation and MRI findings, you have the disease. So that's the symptomatics. And the participants who were considering prodromal, or perhaps some refer to as pre-symptomatic, are those who have confirmed a genetic mutation, MRI findings consistent with ALSP, such as bilateral white matter lesions, corpus callosum thinning. And in this case, we have a limit of maybe 0 to 2. So as long as it's 2 or below, they're considered prodromal. And if it's 3 or more, they're considered symptomatic. Not a whole lot to share about the baseline demographics outside of maybe 3 points. One is that the mean or median age is exactly what's been reported in literature previously, 40 to 45. These folks have had almost a year time to diagnosis into enrolling into the study. And notably, even at baseline, you're seeing a difference in MoCA scores, right? So as a reminder, MoCA score of less than 26, you're starting getting into some mild impairment, cognitive impairment, and we're seeing actually a MoCA score of 20 at baseline. Now into the results. On the left-hand side, we talk about soluble CSF1 findings in the CSF. So as you could imagine, if there's a genetic mutation, you would expect to see less protein production. That's exactly what happens. And in postmortem, you actually find this in tissue. And in this case, we're able to actually find cleaved particles in the CSF. So both symptomatics and prodromals have a profound drop in soluble CSF1R, around 80% or so, compared to healthy controls. Where did we get these healthy controls? Because it's around 51 individuals. So this was actually driven from our phase 1 healthy volunteer study, where we're looking at the pharmacodynamics and pharmacokinetics of the drug. On the right-hand side, I think this topic is quite obvious and is of great interest to everyone in the field, which is neurofilament light chain. So what happens in the CSF and or serum? And again, we're seeing this neurofilament light chain, which is a marker of neuroaxonal injury, for now, at least, showing to be a good marker of disease progression, specifically when you're comparing symptomatics and prodromals, both in CSF and serum. And the good news is that they're also correlate to each other. So hopefully less burden on patients moving forward with us just focusing a little bit more on serum. Now switching to MRI findings. Historically, gray matter atrophy and ventricular enlargement have been flagged as potential or proposed as potential endpoints to look at when it comes to imaging. And in this case, on the left-hand side, gray matter, loss of gray matter is actually evident as early as 6 months. So it's quite profound. At baseline, no differences between prodromals and symptomatics. And within 6 months, you see changes. And the same with ventricular enlargement, maybe a little bit more profound. And that's also, if you look at literature on MRI findings, in addition to corpus callosum thinning, ventricular enlargement has also been a hallmark sign of the disease. So in this case, both at baseline and also as early as 6 months, we're seeing changes. The next 2 slides, now transitioning, I shared with you biomarker data, MRI data. Now we're going to be looking at actual clinical assessments. First, MoCA, and then at the second endpoints, which we'll get into perhaps a little bit less known, but equally important. But when it comes to MoCA, again, having a max score of 30 and 26 below, we'll start to see some mild impairment. At baseline, we're seeing some differences between symptomatics and prodromals, which makes sense. That's probably one of the reasons they were not considered prodromals. And we're seeing a continued cognitive decline among the symptomatics, and perhaps a little bit actually learning how to score better or do the test, specifically with MoCA. So you're seeing a very slight improvement. I would just call it stabilization for practical matters for prodromals. The cortical basal ganglia functional scale, and I don't know how familiar the audience is with this. This was actually suggested to us by the FDA to incorporate. And when a regulatory agency makes a recommendation, you listen, right? And so this is an interesting scale, which it's actually partially failed and completed by caregivers and patients. So it's really nice from that perspective, and also captures all the things that actually go wrong with these individuals, which is motor, which is cognition, and psychiatric issues. The max score is 124. That's the worst score that you could possibly have. So in this case, the higher score, the worse you are. Of course, 0 is perfect scoring. I think this morning I was at a 40 maybe. But overall, my score would not count because they don't just ask you how you're doing today. It's actually they ask you and the caregiver to look back 4 weeks, right? So to make sure that confounders don't influence the individual's response. The last slide, and hopefully we'll have more of this. I'll get to publish this data and a manuscript hopefully probably next year. And this is the things I know when Orchard mentioned, and for practical matters, it's not really confidential to us, but one of the things you would do if you're trying to get an accelerated approval from a drug development perspective is you look at these biomarkers, right, and you try to correlate them with clinical outcomes, and then if the FDA or the EMA or other regulatory agencies agrees, you have a story, right? We are very early stages, but it's pleased to see, at least in this case, when you're looking at correlation of ventricle enlargement with MoCA scores, the larger the ventricles, bottom line, the lower the MoCA scores. So this is the last slide. Certainly we'll be looking to repeat this once over time. So remember, the study is ongoing over 36 months, and also see if we can do the same thing with a cortical basal functional scale score. And hopefully we'll be presenting that in future meetings. So in conclusion, again, as you're looking at the slides, it's MRI and fluid biomarkers are being established as key measures of ALSP pathophysiology. For now, we're seeing a correlation of ventricular enlargement with MoCA scores. And again, it's our gratitude to the investigators, to their teams, and as we always say, that most importantly to the participants. Again, this is a non-interventional trial for a disease with a median survival of 6 to 8 years. So it takes a lot of courage to participate in a study like that. So very, very much grateful to their participation. Thank you for your time and attention, and hopefully we'll be able to address any potential questions.